Well, welcome everyone. Thanks so much for joining us for our second annual Equity and Justice Speaker Series here at NCEAS. Today we have the pleasure of welcoming Christie Bahlai, who is our second speaker in the series this year. If you missed Joan Roughgarden from last month, I encourage you to head over to our YouTube channel where all of our videos from this spring actually and from last year are posted and you can watch those. So I'm Ben Halpern. I'm the director of NCEAS, the National Center for Ecological Analysis and Synthesis. At NCEAS, we work to accelerate scientific discoveries that enhance our understanding of the world and benefit people and nature, as well as to transform the scientific culture to be more open, efficient, and collaborative. So for more information, please check us out at the NCEAS website. Before we begin though, we ask you to join us in acknowledging the Chumash peoples, who are the environmental stewards of the land on which NCEAS is located. So we pay our respects to the Chumash peoples and their elders, past, present, and future, who call this place their home. We acknowledge that this land on which we live and work is the traditional homelands of indigenous peoples, including the Chumash. And this land today is the result of the erasures and exclusions of indigenous peoples. So as we come together to bring awareness to issues of equity and justice, we recognize that the Chumash people and so many other indigenous people are still fighting to dismantle the legacies of settler colonialism in their tribal territories. So as we continue to work towards diversity, equity, inclusion, and justice in data science, we strive to prioritize indigenous data sovereignty and self-determination. I'm going to hand it off now to Heather to introduce you to our speaker for today and to give you some more logistical information about Zoom.
Thanks, Ben, and welcome everybody. We are really excited to have you here. Thank you so much, Christie, for joining us. So just a few logistic items. We are on a webinar, and so if you would like to ask questions, you can do so in the Q&A box, and Christie will be able to see the questions, and so will we. You can ask questions anytime you want during the presentation. You can actually upvote the questions that you really want Christie to answer. You can also put questions in the chat, but we will prioritize questions in the Q&A. We'll do a moderated Q&A at the end of this talk. So now I will welcome Christie Bahlai. She is a computational ecologist at Kent State University, who uses approaches from data science to solve problems in conservation, sustainability, and ecosystem management. She combines a background in physics and, I just did not want to say this word because I knew I would mess it up, organismal? Why is that organismal? It's so hard to say. Ecology, with influences from the tech sector and conservation NGOs to address problems in population ecology. She likes insects, information theory, and practical answers to complex questions. Her current research focuses on developing tools to support information synthesis in temporal ecology. Christie has strong interests in social justice and science and believes that directly addressing diversity issues through technology and culture change benefits both scientists and science. She teaches a course called Reproducible Quantitative Methods that touches on many of these topics. Christie, we are so excited to have you. And because this is a webinar format, you can't hear the resounding applause that would be happening in a normal situation, but I will clap for you, welcome you, and you can go ahead and take it away.
Thanks so much for that introduction, Heather. And thank you, NCEAS, for having me. So my name is Christie Bahlai, and I'm an assistant professor in the Department of Biological Sciences at Kent State University. And I'm really very honored to be part of such a fascinating and important seminar series. Yeah, I've been thinking about this a lot and sort of doing this talk justice. So today I'm going to be talking about some observations that I've made about how fields evolve over time. This is going to be a highly subjective and personal story because, of course, I'm making this call from inside the field. But the interaction of technology and culture and science has fundamentally shaped how my career path has unfolded. And I think it's really important that we think about these things and reflect on them. In science, we like to pretend that we're outside our systems a lot. But that takes both our agency and our responsibility away. So synthesis science is now an absolutely essential part of my personal research culture right now, but it hasn't always been. So what I'm going to talk about today is to try and make the case for why open, inclusive, and synthetic science is not only essential for solving ecological problems, but part of the natural way to progress a field. So one of my biggest interests as a scientist is this idea of time as a driver. Time in and of itself is a really, really tricky driver to reckon with because, well, first of all, most, if not all, ecology has an inherent temporal component. And that's a challenge. If you can come up with something that doesn't, please let's talk, because I cannot think of anything that doesn't in some way depend on time. In most experiments, we're really interested in some sort of temporal question. What's going to happen next? Has this always been this way? Did it change? And then there's the more explicit element. When we're thinking about scaling up processes in ecology, time is a different beast than space, because even in a completely stable abiotic environment, a lot of ecological processes are dependent on internal states, which drive future conditions. And they all have their own inherent scales of operation. But I'm also really interested in this idea of time in the context of when an experiment is done and how that ultimately affects the interpretation of the data. So time can both be an ecological and human concept, and they both affect how the science unfolds. So speaking of time, who I am has changed over time. I haven't always called myself a computational ecologist, and I don't think many people were calling themselves computational ecologists 10 years ago. If you'd asked me then, I would have said I was a population and community ecologist, and no modifiers, and full disclosure, I was absolutely desperate for the community to take me seriously as a capital E ecologist. I was just finishing my PhD in agricultural ecology, and I was working in very simplified systems, agricultural landscapes with single plants planted, and focusing on only a few species at a time. And I was consistently floored by how inadequate the data I was collecting was to actually answer the questions I was asking. You could find some trends, but they were always so variable. And when you looked across sites or across time, you wouldn't see the same patterns. Capital E ecology was this place where people were coming up with these big general rules about how systems operate, and it was where I wanted to be. But to understand how ecology became capital E ecology, we've got to look a little bit at the history of the field itself. So ecology, as we think of it in upper income countries, is actually a pretty young field. It came into its own at the beginning of the 20th century, but underwent a very rapid expansion in high income countries after World War II during the expansion of the American and other university systems. The field of ecology really took off in the late 1950s at a time when American culture was sort of leaning back into this idea of rugged individualism. So let's call this rugged individualism 2. This time it's for capitalism. So we're leaning into this idea of this lone wolf communing with nature. So Morgan Ernest at the University of Florida has referred to this ideal of ecology as the cowboy myth. Now, ecology has always suffered from a bit of an inferiority complex about its place amongst the hard sciences, right? Ernest Rutherford is said to have said, all science is either physics or stamp collecting, which is kind of harsh, Rutherford. Luckily, the field of frequentist statistics had come along at about this time, and it was this way of dealing with scarce data that was messy. Finally, you could take a mess of natural systems data and feed it into really prescriptive equations and find truth. So ecology had to become more rigorous and neat and physics-like if it was to be taken seriously. So how does this cultural priming interact with who's doing the science? A common cliche that we often use is that history is written by the victors, but how do we decide who these victors are exactly? Kirsty Whitaker, who's the program director for tools, practices, and systems at the Alan Turing Institute in London, talks a lot about the role of luck in who gets to break through in science. Essentially, you need both the luck of asking the right culturally congruent questions, but also you need to get the right data to support your investigations. As practicing scientists, we all know that data can be quite variable, especially in small studies. And in fact, this is now a major avenue of my current research, and we'll get back to this. But essentially what Christie is saying here is that there's a strong role of luck in getting that nice, clean, clear data set that supports the story that you're trying to tell. But given that our culture is primed for these clear, simple stories, these are favored. You have to be in the right place at the right time with the right equipment and right supports. And then the victors expect the next generation to move through the field like they did. And the survivorship bias doesn't allow us to see the full range of questions, data, and practitioners of that previous time period. So basically, we get a very limited, homogeneous set of ideas that makes the record, and then the record becomes self-reinforcing. And history has a particular way of remembering our leaders, our influential thinkers. So Sarah Kidway is a historian of science at the University of Rendsburg, and she studies how influential individuals affect how science is conceptualized. She wants us to problematize this lone genius myth, whether it's the stipend from a king, the benefits of status, or just happening to be at the right, hit on the right idea at the right time in the culture that's primed for this idea. All of these factors operating behind the scenes create a situation where one person's work is valorized and remembered and another person's isn't. There's a very human tendency to overcredit our hard work and actions that we've taken, our greatness, if you will, and undercredit the impact of chance, privilege, culture, and being at the right place at the right time. In short, good timing can make you look very clever. So I'm going to show you a classic example from the history of ecology. This right here is Robert Macarthur, and he's famous for, among other things, his work demonstrating that similar organisms occupying the same space will partition into different parts within that habitat, which minimizes competition and allows species to coexist. Macarthur conducted this study on a 9.4 acre parcel of land in Maine in 1956 and 1957. He worked all by his lonesome by observing just how much time 5 different species of warbler occupied different parts of white pine trees. This was an extremely low-tech operation. The first year of the study, Macarthur didn't even have a stopwatch, and he counted how long he observed each bird, 1,000, 1,000. The work resulted in one of the most foundational pieces in community ecology, with a somewhat underselling title, Population Ecology of Some Warblers in Northeastern Coniferous Forests, published in Ecology in 1958. So this and other studies like it became the canon for people practicing ecology today. It became a cultural touchstone, the example of how good, elegant ecology is done. But the thing is, it's also very different from how ecology is done today. For one, not to throw shade or discount the amount of work that Macarthur did, but it's a relatively small scale, small scope study. And I'm certainly not the first to point out that Macarthur's huge influence in the field, and probably not the only one to be a little salty about how the straightforward patterns he observed and why he's remembered for them. But fascinatingly, these nice clean results that have been reported are almost definitely not capturing the authentic variability of this system. The Macarthur warbler study was revisited in 2016 by a graduate student named Bick Wheeler and his advisor, John Anderson at the College of the Atlantic. And in their early published coverage, the patterns weren't nearly as clear as those observed by Macarthur, despite the team having access to better monitoring technology like video recorders and more assistance. And I was really curious about this, and I reached out to Dr. Anderson, who said, and I quote, "Macarthur's work does not stand up very well." So not only did they have trouble replicating any patterns observed by Macarthur, Anderson and Wheeler's team had trouble replicating the methods as described. But nevertheless, this is the field I emerged in when I started my graduate work in 2005. And this is me doing what might be described as a small scale study. Here I am in a growth chamber, culling soybean aphid populations so I could track vital rates. Basically, I wanted to know how populations of these tiny critters could grow on their host plants at different temperatures. And so after spending months carefully rearing plants, watching aphid eggs day and night until they hatched, and moving around my little buddies, on the plants without ripping their faces off, which is harder than it looks, I developed a model. And the model that I developed worked great in the lab. Air temperature was all we needed to predict when the aphid eggs would hatch, but it didn't work in the field. And that was really deflating for a new grad student. It made me wonder if I was sciencing right. And it was my first lesson in how the field environment was a lot more complicated than the lab. And of course, I'm not the first grad student to be thrown off by the failure to reproduce work or translate it to another system. Nicole Nelson, a professor of science and technology studies at the University of Wisconsin-Madison, specializes in how scientists conceptualize their own work within their understanding of the natural world. She's currently working on a study examining how graduate students respond when they first encounter irreproducibility within their own work. When we spoke, she told me that a lot of students start from a place of thinking that the literature is infallible, that the natural world is this regular, repeatable thing. And so if they follow the steps, they should get the right answer. And if they don't get the right answer, they turn to self-blame. Now, a lot of us look at this bewilderment as a rite of passage for students, and to a certain extent, it probably is. But the implications are a little bit more insidious than that. When we consider the context of this system that's reinforcing homogeneity in scientists, graduate students who are different from the heroes of the field are already getting messages from the culture that they might not fit, and this will exacerbate the problem. And this signaling plays out in who gets to participate in and who gets retained in science, who is or who is not pushed out. So Jason Williams is the Assistant Director of Inclusion and Research Readiness at Cold Spring Harbor Laboratory. And when we spoke about his work, he explained that the image of infallibility and rigidity was really alienating to science learners, especially minoritized students who weren't shown the journey of science, but instead this very clear-cut, curated destination. He emphasized the importance of modeling, not knowing, trying several different possible solutions, and even failure when training new students in scientific skills. Jason also talked about how the standards of evidence, essentially what we need in order to show a hypothesis is supported, it grows over time. Data itself gets more extensive and messier. And so there's this critical need for multiple perspectives, a community of perspectives on data. So we get this critical intersection of increasing data and increased need for robust, diverse approaches, coupled with this system that pushes people out. I started to become really interested in this increasingly complex world of data and analytics and how dependent the process of finding out was on who was making the decisions when I was still in grad school. First, it came from my own work. The data I would collect wasn't enough to make generalities. It seemed like the more data I collected, in fact, more caveats emerged. And then when I tried to find generality in what I did, I'd write a paper and I'd submit it only to have reviewers comment that my findings were better suited to a small regional journal. Ouch. But they weren't wrong. How would I know if my findings didn't apply to organisms in other spaces? Even with my own studies, data were really variable. Then when I helped other people analyze their data, it was more of the same. I had a feeling of uneasiness that seemed to center around the statistics that I was trained to use to solve these problems. They gave a binary answer. This is significant or it's not. So it's true or it's not. But truth was very sensitive to the data that I was feeding and really sensitive to the structure of the experiment. So science was disconcertingly human. So I'm actually going to illustrate this phenomenon of how we're asking questions and the answers we're getting being dependent on who and when and how we're asking them with an example from my field, which means I'm going to talk about insects, specifically the case study of what's going on with global insect populations. So you've probably heard at this point about several high-profile studies that document the insect decline phenomenon. Now, this is one of those superficially simple problems that unfortunately is a lot more complicated than how it's being portrayed, both within the media and a lot of higher profile scientific literature. Insect dynamics are, in a word, sensitive. The number of insects can vary by orders of magnitude from generation to generation, time to time, and place to place. They're fecund, they have a lot of babies, and they're also generally pretty short-lived. They're remarkably sensitive to environmental conditions and resource availability. And even in completely stable environments, they're prone to boom-bust dynamics. But the conclusions we reach are also remarkably affected by simple decisions in how people are choosing to monitor them. So for example, the frequency that we sample a system can really strongly affect our interpretations about the system. When we perform a statistical analysis, we're abstracting, right? The abstraction is necessary, but sometimes these decisions lead us to make simple conclusions that seem really obvious and unquestionable when you look at the data in a particular way. So here's a plot inspired by how the results were presented in one of the studies documenting insect decline. A survey was done in 1977, and then the author went back in 2013 and resurveyed and found fewer insects. But what if we looked at the data a little bit differently? What if we were able to monitor more frequently? Whereas before we had 2 snapshots neatly summarized, here we capture more of the variability in time. You can see here that the patterns represented are actually statistically the same in this hypothetical data set. The bar plot is not untrue, but we probably feel a little bit differently when we look at this data presentation. We can see the variability better and would probably feel a little less comfortable making a linear prediction because in this case, the data feels a lot less directed. Increasing how much we sample makes the data feel messier. Life is easier and probably more publishable when we just had 2 data points. But it gets worse. The tricky thing is when you start sampling can also matter an awful lot. So where and when you choose to measure can make a huge difference about what you find. And this concept is illustrated beautifully in a recent paper by Fernier, White, and Hurd. So I want you to imagine 2 populations of dragonflies in 2 ponds, and both of these populations are undergoing identical dynamics, but they're out of sync with each other. Now imagine you only have the resources to monitor one of the populations. You're probably going to choose the site where there's more dragonflies at the start of the study because it's better for dragonflies, at least by your measurement at that time. The problem, though, is that if you capture a population at the peak of its cycle, it's nearly statistically impossible to, upon going back and completing the survey again, get anything that doesn't look like a decline. It makes understanding the underlying variability of the population in both time and space absolutely essential before you can make any inferences at all. So as you can see, the context and the perspective or window that you're looking through matters a lot. And this is just for the simple factor of when we do our research. So really, given these conditions, it's no wonder that scientists have failed to come to a consensus on the direction, magnitude, and drivers of global insect decline. It's not even clear we're asking the same question. Clearly, to solve things like the biodiversity crisis, we need to put our heads together. And lucky for us, this is, to an extent, happening. I think we are, in fact, in a cultural transition where our field is starting to recognize a more collectivist set of interests that help align our approaches to science with more collective good in mind. Ecology as a field has really, in the past decades, been investing in this idea of synthesis, bringing together diverse data, perspectives, and ideas to help solve these bigger societal problems. Between the connectivity of society driven by the internet, the recognition of global scale environmental problems that require urgent action, and the increasing automation of environmental data collection, it's not just possible, but essential to look at ecology at new and larger scales. The technology enabled the change, but the culture of connectivity wrought by the internet, in my opinion, is what primed the individual scientists to be receptive to these bigger changes. And so those came a little bit later. Now, this was before my time as an active participant in the scientific enterprise, but it appears the idea of synthesis started gaining momentum in the mid-1990s with the formation of NCEAS. And started really gaining popularity in the early 2010s with SYNC and IDIV both being established in 2012. But the thing is, for the goals of synthesis to really be realized, another shift needs to occur in ecology, this movement towards open, interoperable, and shared data. So in 2012, comes up as a popular number, when I finished my PhD and started a postdoc, I signed on to a cool project at Michigan State that I thought would be mostly looking at niche partitioning in an invaded community of ladybugs, like Macarthur, but with ladybugs. What I didn't realize at the time was I was going to be introduced to a completely different way of actually doing science, networked science. The Ladybug project was part of what was called the main cropping system experiment at Kellogg Biological Station. Kellogg isn't just another research station. In 1989, they'd established this experiment as infrastructure to support interdisciplinary science, to study how processes in ecological systems unfold over time. At this site, interdisciplinary teams went beyond occasional collaboration. They had long-standing scientific relationships built on understanding interactions within the systems and how they unfold over years and years and years. To make this work, they needed to break down the disciplinary silos and work together to maintain experiments, share data, and really, really deeply interact with each other's work. What was more, Kellogg was part, although it is a unique and special place, but it's part of a multi-site network, the long-term ecological research network of 27, it's 26 or 27 sites where groups of interdisciplinary scientists are doing place-based network research like at Kellogg. So I still count my lucky stars every day that I landed in such a fertile research environment for my postdoc. And it gave me this opportunity to make the questions that I was asking much bigger. Scientists in the network paid special attention to data management, analysis, reproducibility, and collaboration, because with a long-term experiment, the data you collect is not yours. It belongs to the experiment, and it was going before you got there and will likely continue after you leave. You're a piece of the puzzle, and you have to work with each other to make this bigger picture. Despite being embedded in this individualist landscape of academic scientists, the LTR sites had learned to foster this collectivist attitude of investing in culture. Yet, almost ironically, the network has fostered the careers of some of the most prominent ecologists of our time, perhaps because they aim to invest in and promote people who act as stewards of the science and the collective good. This is the environment that I came to define myself as a computational ecologist in. This, as a person who had my own work, but also supported others in analyses and bridges together data and people. So this is where I learned that the best science is done with collective in mind. And it was in this environment that I got the idea that we could, in fact, formally study the effect of being in the right place at the right time, or wrong time, depending on and its effect on scientific conclusions. So this figure was built with several Michigan State students during our class project from my reproducible research course. So we used long-term data examining firefly populations out of Kellogg Biological Station. And we were mostly interested in within-season phenology. But the students had some really compelling observations about this particular figure when we look at the population dynamics over our entire study period from 2004 through 2016. Namely, the observation period seemed to have a really strong impact on what conclusions we would make about the system. So for instance, let's see there. What would happen if a master's student came in 2004 and monitored for 3 seasons? What if a PhD student started in 2007? What would each of those students conclude at the end of their study? So what I did was I built a data mining tool that essentially automates our what ifs. It takes time series data and applies statistics in an objectively terrible way by resampling the data, performing a regression, and collecting the statistics at every possible observation interval. Think of it as thousands of automated, re-randomized graduate students applying the statistical analyses they learned in their intro stats class faithfully over and over and over again and compiling the results. And it allows us to ask the question, how likely is it that we got the timing wrong in our shorter term study? And so what do we find when we look at data this way? Well, misleading trends are everywhere. So to orient you to this plot, each of the dots is a conclusion, the magnitude of a slope that we'd reach about the system and its behavior over different sampling periods. The size of the point is our statistical confidence. In this case, we're scaled by R squared. So you can see that there's a lot of difference of opinion. Basically, you can get any slope, but also a lot of confidence in our divergent answers during shorter study periods. For the Firefly data, we concluded that our digital graduate students would rarely agree on how the system was behaving unless we had them collecting data for more than 6 years. The role of chance in our conclusions is bigger than our statistical approach would like us to think when temporal processes are involved. Now, this is an obvious finding. Data that's linked in time is non-independent. So we are violating the assumptions of our linear regression, but that doesn't mean that this approach isn't being used in science and used frequently. So with a group of colleagues led by Sarah Cusser, who joined as a postdoc at Michigan State, just as I was packing up and leaving to start as new faculty, we became interested in how this phenomenon might affect decisions and recommendations that we were making in applied systems. So we used a 30-year data set on the performance of no-till field crop agriculture from the KBS site. And we asked the question, how likely are we to see costs recouped over different observation periods, looking at no-till versus conventional tillage? Now, we're generally in a place now where there's a fairly good consensus on no-till approaches for field crops, but this was a controversial intervention in its time. And what we found was that evidence of why this controversy existed. That is, in situations with less than 10 years of data, almost a third of all trials would produce a loss instead of a gain. This is one site under consistent management for the entire study period. So no wonder shorter term studies took a while to converge on an answer. When Sarah applied this approach to other population time series data, specifically, we harvested 100 time series more than 12 years in length, where press disturbances were applied to populations in the whole LTR network. This time, and included data from Antarctica all the way to Alaska, we found that the minimum time needed to make observations varied a lot. And in fact, it took longer to be able to observe a stable trend in variable environments. Plant community data was particularly sensitive to this environmental effect. So what does that mean? It means that the take home is that changing and changeable climates means it's going to be even harder to make predictions about our system. The when of the research matters even more in these systems. So we can see that how we ask the questions matters a lot when we're talking about what kind of conclusions we'll make. And so something even as simple as when we're looking at a system can fundamentally change what we see, which leads to this critical imperative. Without diverse perspectives, without incorporating more windows of view into our science, and without meaningful inclusion, the science itself will suffer by definition. Rather than becoming an arbiter of truth, the science itself becomes biased by its own homogeneity, or worse yet, outright misleads us. In order for science to thrive and to expand and answer complex, multifaceted problems, these heterogeneities, the human messiness that seemed like bugs in our current system, are actually features. Dr. Leon Walls, a professor of education and social services at the University of Vermont, has found that the context and perspective have fundamental implications for the results of research. Sometimes when using the standard method or the easy sample, like university students, the story will leave out really important information or can become unnecessarily cumbersome and potentially delay action or help to a community that needs it. Often it takes a member of the community to help reframe the research in a direction that's relevant and actionable. And sometimes to zoom in and look for those fine-grained details and variabilities is essential before we can move on to making generalities. So in my opinion, the cultural shift towards networked, open, and synthesis science creates an opportunity to make what Leon's saying a reality, connecting the silos. Let's see what we've been missing and shine a light on the true variability of our systems. And there seems to be cultural support for this movement as well. So collaborative and synthetic and data-driven model of science is not as fringe as it once was. Increasingly, journals are requiring scientists to share their data, and major funding agencies now all over the world have required data and code sharing. This all really formed the idea of the work that I wanted to do as I built my own research program. But a little bit of a rosy view, I'd underestimated cultural inertia. Shall we say it's the lagging responses to disturbances, if you will? So when I hit the academic job market in the mid-20-teens, I went out there with the full confidence that open science and data-driven discovery were my calling, and I was going to have a career in computation and synthesis of heterogeneous data. But the dominant academic culture and the attitudes of the people in power, they had programs shaped by the cowboys of yesteryear. And they didn't see how what I was proposing to do as a research program could possibly be viable. So in fact, this came to sharp relief during my very, very first faculty job interview. So those of you in academia know how these interviews go. They are trial by fire marathons where you give a research seminar, have a series of interviews with faculty, deans, human resources personnel, the works over a period of a couple days. During my exit meeting, after explaining my research plan, once again, the search committee chair paused, cleared his throat and said, "Well, that's all fine and good, Christie, but what happens to your research when the data runs out?" Taken a bit aback, I said, "Well, if the data runs out, I'm sure my research program will be the least of our problems." I didn't get that job. So I think that we're in this really critical period here. We're in this place where the cultural shift is occurring, but we need to be conscientious about how we go about it. There's a temptation to define the successes of big and open ecology in the context of the previous paradigm because these values are still obviously very much in play. To sell the collectivist approaches as a means of advancing ourselves, advancing individualism. So papers with open data get cited more, reduce delays in producing more papers with your open workflows, increase your influence, all data can be yours, increase your power. But this leans into values that ostensibly we're trying to redress when we get to bigger, more inclusive spaces that welcome variability that is innate to holistic examination of heterogeneous systems. Like colonists coming to conquer new lands, open data advocates in the dominant paradigm often frame data-driven discovery with the same language of the frontier myth used on lands. Data is the new oil, anyone? But where is the data coming from and who is impacted by its extraction? Like other forms of settler colonialism, in many cases, there were already people there. What if instead of extraction, we talk about data sovereignty? Dr. DeAndre Smiles is an assistant professor in the Department of Geography at the University of Victoria in BC, Canada. And he focuses on critical Indigenous geographies, human environment interactions, and political ecology. And he wants research to move from this extractive model to a collaborative one. He's written extensively on settler colonialist ideas that frame non-living entities from bodies to outer space to data as things to be extracted. But what about accountability and stewardship in these spaces? Open science and open data isn't the end of the story. Researchers need to be active in their collaborations with communities. The hierarchical model, with its lone geniuses, looks at collaboration as a means to an end, a great way to get publications out the door. And open science isn't immune to this way of thinking. But data comes from places and people, and we've seen again and again and again that that's not neutral. So how do you use power once you've got it and use it for good? I think that within all of our fields, there's some version of cowboy ecology. The values and structures of the past, which somehow both shape our scientific culture and hold them back from being able to do the things we value. It requires constant intentional vigilance to direct cultural change for something better. It requires investment and I think we put a lot of pressure on students and people who hold the most precarious positions in the academy to be the change. And I don't think that's a fair thing to do. We've all heard calls to action where we say we need more education for our trainees and we need the young people to step up. And I think that's great, but it lets late career folks off the hook more than I'd like. In a culture where people whose identities are least likely to fit or be regarded as newcomers, they are most likely to be dismissed when they suggest changes to practice or value, and it costs them the most. It's essential to have these voices both heard and actively advocated for all of those of us who have gained whatever modicum of power we have in the system. So with that, I call to my fellow, let's say early mid-career folk. We have a huge responsibility to advocate and pave the way for open and inclusive scientific practices. Ecology is now in a place where this work is not fringe, and that gives us a platform. So if you have a platform, what good can you do with it? So I'm going to close with a little bit of data. So when can we be confident that we've taken in all the variability and we've integrated enough context? Well, after 30 years of continuous funding from the NSF, an estimated 100 field assistants, 2 generations of project PIs, continuous environmental monitoring, over 40,000 specimens collected, I think I'm finally comfortable saying that there's probably some spatiotemporal niche partitioning going on between at least 2 of the species of lady beetles that I set out to study in 2012. The 7-spotted lady beetles uses the landscape earlier in the growing season and tends to do better in seasons that are cooler and drier than the multicolored Asian lady beetle. So boom, there you go. Finally, some Macarthurian science coming from the Bahlai lab. So to close, in ecology, we are in no danger of the data running out, but we do have to be mindful stewards of it and the culture of our field to help maximize its potential. I think that the world would be a better place if we all spent more time thinking about and being thoughtful about the data. And it can and should vary. Clear, simple patterns that emerge from complex systems are a signal that we are not capturing authentic variability. Data isn't neutral. It comes from us, and we're messy creatures living in a messy world. This non-neutrality isn't inherently a bad thing, but it is an important thing. What we choose to do in our work and our lives matter. So getting some perspective and contextualizing our understanding in a situation is so important if we're going to solve big multidimensional problems like climate change and biodiversity loss. I think the key to this is embracing variability in data, perspectives, and people in an authentic and inclusive open approach, and keep fighting against the cowboys. Thanks.
Thank you so much, Christie. I wish this could have been live. You had me smiling, you had me laughing, you had me saying, amen, sister friend. Like, probably you're speaking to my choir, but I just thought this was absolutely fabulous. Thank you so much for that. So we're getting a lot of great feedback in the chat, but if anyone does have a question, feel free to add it into the Q&A section and we will address them as they come up. Oh, sorry, I just got distracted by the chat. While people are potentially thinking about their questions, one thing that kind of struck me or that I was noodling on as you were talking is I love this idea of kind of it's showing the journey of science and showing that failure does happen. And, but then it also, with COVID and with science and with the public view of science and how there's this idea that a lot of people don't understand how science is done. And I think there's a fear that if we show what actually is happening, then maybe people will have less confidence in science. Or it could go the other way. Maybe if we actually start normalizing that we're showing that, what science is, then maybe we can build confidence. But I'm just wondering, could you kind of talk about how we can achieve both those objectives of showing what's under the hood, but also instilling confidence in scientific findings?
That is a really awesome and really, really difficult question.
I just thought I'd start with an easy one.
Well, so I've talked a lot with people about this exact concern. And so like a lot of, when I was doing a lot of my open science advocacy during my postdoc, I did a fair bit. I would often get this question from the folks that I was trying to convince. It's like, are you trying to show that we do not know what we're doing? And I go, well, not exactly. What I'm trying to show is we often see in the discourse that, we see things like scientists don't know what they're talking about because they made this recommendation and then it changed. And, any practicing scientist goes, well, of course the recommendation changed. We reevaluated what our recommendation was after we collected more evidence. And like, that's a sign of healthy science that, well, like we know that red wine and chocolate does or does not cause cancer because it's probably a weak effect and it's probably going back and forth quite a bit. And so these recalibrations are actually a sign of the health of the field. And so I think that part of the problem is we've got sort of this idea of science as a set of facts. And maybe we need to teach earlier that science is a process-based way of knowing instead of, you need to memorize Krebs cycle kind of approach. And I mean, I think back to my own early science training and, things like memorizing the periodic table and doing an experiment and getting the wrong answer and then being told, well, here's the right answer. This is what you should have got, rather than trying to interpret what we found. And I know there are a lot of people who are doing this process-based work. So one of my colleagues, Bridget Mulvey, she studies STEM identity. So how people come to identify as scientists and learners in young children. So she does early childhood education up through high school and college students. And so she really advocates for this process-based teaching. But part of the problem is this cultural inertia. We have sort of this lone genius idea where we have, well, the genius found this, so this is the truth, and this is this immalleable concept. And so, yeah, so I am rambling and going in circles because I do not have a good answer here. I think that it's all about creating a culture of science as a process rather than science as a collection of facts.
Yes. Yeah. We don't have any other chats, any questions, so I will encourage people. There's Q&A box. If you have anything, if anything struck a chord, feel free to add your question in there. I think another part of your talk that I was amening to was really around this idea of the new people to the party having to be the advocate for the open data. And so I guess I'm wondering like how we can start to merge those two together. How can we get the old guard, so to speak, more interested in this process or open to this process, or maybe those aren't the right words. I'm just not quite sure. How do we start to bridge that divide from people that have less to lose if they stick their necks out a little bit?
So, okay, this is my lawful good interpretation of this, but I think that funding agencies and regulatory environments are going to be the factor that affects change sort of at the cultural level. Because, well, okay, this is personal experience, this is anecdotal data, but I remember having sort of side eyes at me through all of my open advocacy. And then in 2016, NSF published their Today's Data, Tomorrow's Discoveries guide and created this requirement for data management plans. And suddenly the folks who were like, well, I've always gotten NSF funding before. My whole career has been funded by National Science Foundation. And now they're requiring this new thing from me. And it wasn't a happy thing but it was I guess this is a thing I have to do now. And suddenly people started calling me back and saying, hey, okay, so how do I do this now?
Yeah.
And so I think that, I hate to use the carrot and stick analogy, but not necessarily a stick, but the lines on the side of the road, creating those guidelines. You can still drive over them, but you're probably not going to get as far as you want to if you don't follow sort of the regulations.
Yeah, thank you. So we do have some questions coming in now. Our first one is from Hannah, and she says, as students, how can we try to accommodate the messiness of our data and the systems we study while still managing to actually publish the early work we need in order to be competitive as researchers and in the job market?
Yeah, that is hard. I think that, there are people who have learned ways to talk about messy data elegantly, and they're the ones who get the high profile publications. And I would love to be able to say, we don't need high profile publications, your work is valid. But I'm also acutely aware, I'm going up for tenure this year, acutely aware of this need to show productivity based on traditional metrics. You need to be able to do that to survive in order to get the power you need to make the change, right? And so there's this conflict. I think that being okay with publishing sort of messy stories and convincing folks that we shouldn't put that data story that doesn't tell the cleanest thing into the file drawer. We should probably publish a paper that may not go to a fancy journal, but gets out there in the record. That's a good step. But yeah, there is that conflict. I actually gave a talk at a plant sciences symposium with a bunch of agricultural grad students, predominantly agricultural grad students last week. And the question was like, well, how we've got these systems that, you do 2 years and they're completely different from each other. What do you do? And I said, well, go for the third year. That's the tiebreaker year. That's what we always called it in grad school, is the third year is the tiebreaker year. But it is hard, like trying to figure out that story and figure out how to tell the story when acknowledging that the data is really very variable. Yeah. I often feel a lot of conflict about this. So like my students had to change experimental plans over the pandemic. And so their data they collected, smaller data sets and more variable methods than you would have ideally hoped for. And so the trends we see are pretty messy. And well, we're doing our best to explain that variability and just authentically present, this is what we saw when we did this. And that is not a satisfying answer, because I fully acknowledge, having that publication record is what is evaluated when we're engaging in academic science. Yeah. People are really impressed with software, though. If you start putting software products out, I find that really impresses the old guard, because they're like, oh, well, software. Okay, cool. So see if you can write something that analyzes that messy data and shows how you can get that story together.
Awesome, thanks. We have a question in the chat from Marty, Marty Downs of the LTER, and she mentioned 28 LTER sites. So,
oh my, wow.
Yeah. And so she writes, I love, of course, your take on LTER scientists as stewards of the data. From your now inside and outside of LTER experience, what are you thinking about how to better cultivate that attitude more broadly?
This is hard. This is why I desperately maintain my connection to the LTER network as hard as I can. Because once you sort of get out of that networked science approach, everyone sort of has their own benchmarks. And when you have the collective benchmarks, it really, really helps with fostering that collaboration on sort of a long-term permanent basis. And so LTR is not the only organization that does this. I'm part of a project here called the H2 Ohio project, which is wetlands monitoring associated with the state of Ohio. And there's meetings, and the research direction is led by my wonderful colleague, Lauren Kingsman Costello, and she's doing everything she can she's also a graduate of the LTR program, by the way. And she's doing everything that's possible to try and maintain this multidisciplinary view on maintaining the research. And I'm really excited about that project because that particular ethos is being brought to it. Part of the problem, though, is these things cost money and these things cost resources. And so we don't have time to maintain things like this if we don't have funding to do that. And so these are rare and beautiful things. And I think that every scientist should be involved in some sort of network project. But I recognize that resources can sometimes limit that.
Well, I think this whole talk could be a call to funding for synthesis science and in general. So thank you so much. With that, I am going to end it. Ben, did you want to say something? No, okay, I saw you pop up. But thank you so much for joining us. I really enjoyed the talk, and we will hope to continue to work with you in the future, but thank you so much and good luck with tenure.
Oh, thank you so much. Thanks so much for having me.
Bye-bye.